plate, as we can see in here, right? And one part will be the brain and the rest will be the spinal cord. The cells that form this neural plate, as we see in these two sections, are neuroepithelial cells, okay? And they will form through a fantastic transformation that needs to lengthen and then to form a tube, okay? And then, as we see in this image, so it starts from a flat structure that was the last section that we saw previous to reading week, right? Into folding and creating the brain structure. This is the head process, and then the spinal cord. So during this fantastic transformation, the ectoderm will form the epidermis and the nervous system, both at the same time, and all is coming from the ectoderm, okay? So a portion of the dorsal ectoderm will be specified to become neural ectoderm, which will form this neural plate that we see in here. And the cell is going to start becoming distinguished. And this is because they're going to start attaining a columnar shape, okay? So we see here they start lifting, and then we're going to finish with a column and and the tube formation, okay? So the neural plate becomes the neural tube through a process that we're going to be looking at, which is called neurolation. And the neural tube, as we know, is the rudiment of the central nervous system, okay? So an embryo that is undergoing neurolation is called neurula. So in this image, we see the different aspects that we will be covering within the next 2, maybe 3 weeks of lecture, okay? So we will be studying how the most complex system of the vertebrates form, which is the nervous system. It's very hierarchical. Every cell plays different roles. Every group of cells play different roles, so we can do the multiple tasks we have right now accomplishing. So as we can see in this figure, we will start with the formation of the neural tube. So this is our neural tube over here, okay? So we start with a flat. Remember when we finished during gastrulation, we ended up with one single layer on the outside being the ectoderm. So this is the NT, this is the neural tube. So we start from here, and then it's to transform to become a tube, okay? So we start that flat epithelium, then transform into different type of cells, changing the form. Some of the cells will become a little more of the neural type of cells. So they want to change a little bit the fate because they were all part of this neural tube, right? But then they need to become different type of cells. So here we're going to have cells that are going to follow different patterns. And then some others we're going to transform and going to go to this epithelial mesenchymal cell transition, okay? So flat epithelium into a completely different structure, as we can see, cell fate specification as well. It all depends on what time they separate from this epithelium. And we're going to be calling that the birthday of these cells. So your neural cells have different birthdays at the time that they start migrating, getting away from mama. That is this epithelium that was at the beginning. So this is a very delicate orchestra, all working together in harmony to create the hierarchy that we're going to be seeing here into the neural tube and the nervous system. Some cells going to be close to the lumen of the neural tube, and some cells will be farther away from that lumen. And this is what we are being covering in this chapter. So from that single layer, flat layer, plate, okay, of the epidermis, we're going to have different group of cells. So the ectoderm is the surface of the epidermis. Okay, will constitute the larger organs of the vertebrates. The epidermis is the largest organ that we have. Then one section will become the neural tube. Okay, and then neural tube, we're going to see, have the brain, neural pituitary gland, the spinal cord, motor neurons, and the retina. So all the pigmented part that make the melanocytes and so on. But we're also going to have the neural plate. Okay, so that neural plate, as we're going to see later, will become that neural tube later on. So we're going to have different molecules, and in this case, we're going to be talking about the BMPs, bone morphogenic molecules, that they're going to be playing a role into the specification of the cells. Once again, we're going to have a gradient, as we're going to be seeing. Okay, so if we have low levels of BMPs, cells will be specified to become the neural plate. So low amounts of BMP, the cells will become the neural plate. Intermediate levels of BMP, then you want to induce the neural crest cells. So these ones here, so this is where we have a moderate levels of BMP. So these are the ones that are going to specify those cells to become peripheral nervous system cells, the adrenal, melanocytes, the facial cartilage, and the dentin in the teeth. Okay, but high levels of BMPs will induce to become a little bit more of the epidermis. So high levels of BMP are the epidermis and all the derivatives that we see. So here we see how just from that flat neural plate, as cells start migrating and changing their shape, they're going to be subjected to different concentrations of BMP. High concentration, it will be epidermal and derivatives. Medium concentration, neural crest cells, and then as here, low concentrations, we're going to have the neural tube. Is this clear? How the BMP will affect this? Okay. Okay, so in this figure, we see the primary neurolation in amphibians, and we're going to be comparing these ones a little bit too, okay, between amphibians, mammals, and birds. So the three portions of the ectoderm, which means the surface ectoderm, the neural crest cells, and the neural plate tube, are differentiated through that process that we call neurolation. Okay, so in here, this is the dorsal surface of this neurula in amphibian. So we have the neural plate surrounded what we knew as the groove, right? And the neural fold this is what it has a different color on the outside. Okay, so we see this in the early neurula, mid neurula, and late neurula. So the neural plate is what I want to attract your attention right now in here in the purplish color. So after gastrulation, where the node went all the way up here, we have the pit here, okay, and the blastopore over here at the back, going to start transforming, and the transformation starts here at the neural plate. So in that transvex section, here in purple, that is the neural plate, and surrounding that is the neural fold. We're going to see that this plate start elevating. This is what it's doing here, start elevating, and then joining at the top part in the late gastrula. So now wherever it was in the surface gets inside and will be covered completely by that neural fold, okay? So the process of neurolation requires the expression of different transcription factors. Most of these ones are SOX. SOX 1, SOX 2, and SOX 3. Okay, so this transcription factor, SOX 1, 2, 3, do two things that are very important. The first one is that activate the genes that specify cells to become the neural plate. Okay. The second one is that it inhibits the formation of the epidermis on the neural crest, because we know that most of this sector will become the epidermis, right? But these cells in the middle, in the neural plate, needs to remain ectodermal, so they'll become neuroectodermis, okay? So neuroectoderm, excuse me. So we're gonna block the signaling, and then we're gonna block the signal of the BMPs. Okay, so remember that this requires low BMP, right? We go back to here. To become the neural tube, we need low levels of BMP. So we need this SOX 1, 2, 3 to block the signal of the BMP, so it is down-regulated those BMPs, okay? So this always brings me to remind you guys how some signals can promote the specification of one type of cells while inhibiting the specification of others, okay? So some subjects that we discussed at the very beginning of this course. So these SOX transcription factors establish the neural plate cells as neural precursors for the formation of the nervous system cells, okay? So as we can see, this transformation goes from the gastrula through the neural, and is in the neural plate, and in order to, at the end, have that neural tube form in here in the middle of the embryo. So there are two major ways to convert the neural plate into the neural tube. And those two is primary and secondary neurolation. And please pay attention to this, because once we get into the lab, sometimes this becomes a little confusing, okay? So in the primary neurolation, the cells surrounding the neural plate direct to the neural plate cells to proliferate. They will invaginate, and they will pinch off from the surface to form a hollow tube, okay? So they proliferate, invaginate, and pinch off to form the tube. In the secondary neurolation, the neural tube arises from a solid core of mesenchymal cells, okay? So we don't wanna see the elevation, joining, and separation. This is a solid core that is placed in the middle, okay? And normally is seen in the most posterior region of the embryo, okay? So the complete neural tube formed by joining these two separately. So we're gonna have primary neurolation at front, secondary neurolation and the second half of the embryo, okay? So together. So in this one, we see clearly this primary secondary neurolation. So in the anterior portion, as I said it is primary neurolation. And the posterior portion, then it is the secondary neurolation. And in the middle, of course, we have a junctional neurolation, okay, that combine allows the fusion of the two, okay? So the colors are indicating the fate of the cells, okay? And what we see in here, the ECM that we see in here is simply the extracellular matrix, okay? So we see the neural plate ectoderm here in the middle in blue. Then we have the paraxial mesoderm on the side, and starting from the notochord, that is structure that we're going to see again and again as inducing a lot of things into this embryo. And then in yellow, we have the extracellular matrix at the base with more ectoderm. Okay. So from an evolutionary point of view, it seems that the original process of neurolation was primary neurolation. Okay. This is the original. Okay. The secondary neurolation appears as an innovation to allow the appearance of limbs and tail elongation. Okay, so birds will have primary neurolation that generates the tube anterior to the limbs, to the hind limbs. Okay, in mammals, secondary neurolation occurs at the level of the sacral vertebra of the tail. And in fish and amphibians, the tail neural tube is made by secondary neurolation. Everything else is primary neurolation. Is this concept clear? So primary neurolation for anything that is above the tail, okay, and anything that goes below the tail, like in limbs, then it becomes secondary neurolation. Primary neurolation, we have the formation of a tube that pinches off and get as a separate structure. In the secondary neurolation is a hollowing of a cord that occurs. And then between both of them, we have the junctional neurolation, that is the transition zone, that has a little bit of each of them, okay? Okay, so now let's go through the primary neurolation process. And drink a little bit of tea. So, primary neurolation, the ectoderm is divided into 3 sets of cells. Okay? The nearest part of the sagittal plane will give rise to the neural tube, and from which we're going to see the brain in the anterior portion and part of the spinal cord that will raise from that section. Okay. The most lateral, so we're looking here at the lateral section over here and over here of the ectoderm will constitute the epidermis, right? Because once it's closed, this gonna pinch off and this will become the epidermis. So the mid-sagittal section will be the brain and the spinal cord. The lateral side region of that ectodermis will become the epidermis. Now, the neural crest cells form in the region that becomes the connection, the neural tube and the epidermis. So somewhere in here, let me see if I can bring these ones. You see these little red dots in here? Those are the crest, the neural crest cells that are gonna go through the process of epithelial mesenchymal transformation. And it's always there at the junction of the neural tube and the epidermis, okay? So we're gonna have, from those cells, we're gonna have some of the neurons and the glia cells, okay? Pigment cells of the skins, all of them gonna come from there, those neural crest cells. So let's see the process. So this goes a summary. This is a summary of the primary neurolation, okay? So this is our neural plate. On the edges, we have the presumptive neural crest cells, and at the exterior, we have the epidermis. What will become epidermis eventually is also presumptive. So we're going to have an elongation first, okay? And this occurs at the anterior portion or the rostral caudal portion, okay? So at the anterior portion, and it moves to the back. So shortly after the neural plates had formed, So just at the very end of gastrulation, beginning of neurolation, okay, we're going to see a little bit of a thicken and bending upwards. So we see here the thickening, right? It's not just flat, flat, flat. We see a thickening that allows the elongation and a rising up, that is the bend, okay? This is the elevation that is occurring here, okay? And this is to form what we're going to be calling the neural folds, okay? So that's why we call it the folding, okay? So we have elongation first to thicken that section, and then it's going to be raising up to form the fold. So that's the neural fold. So it has a little bit of a U shape, right? Inverse U in this section and this section. And in the middle is what we're going to call the neural groove, okay? So it appears just there in the middle, a little bit with an orange color in there. And that will divide the left and the right sides of the embryo, okay? As that passes by, this continue to elongate, continue to fold in here. And we see here the MHP, that's the median hinge point, okay? That is like a hinge on a door, right? You can open and close. So that's the mid-hinge point, just in the groove, okay? Right in the middle, separating the right and the left side, okay? So we continue with the elevation of the neural folds in here. We see that this mid-hinge point, the cells start having a great transformation. That is what we see here, start elongating to allow the sides to continue going up. Those neural folds continue to migrate to the midline of the embryo until they converge. And this is the convergence point, okay? So whatever we have here as the presumptive neural crest in here and here are joined in this section, okay? And the point that they joined, gonna start fusing, but here, I want to call your attention to this point. Oops, nope, that's not what I wanted to do. Right here. This is the ectoderm that joins that neural plate cells, right? Right in where they converge, we want to see the appearance of the dorsal lateral hinge point, or the DLHP, okay? So this dorsal lateral hinge point anchor to the surface ectoderm that will become the epidermis right here, okay? And the overlaying ectoderm, okay? So here, all of this, we knew that it was the neural ectoderm, right? Now, we can observe that the cells at the top of the neural fold start delaminating and become neural crest cells. So if we come here, that point of conversion, right, we see some cells that start delaminating, so losing the connection to the tissue, okay? And these are the neural crest cells. Now, at the very end, what do we have? We have the cells on the top are the epidermis, we have the formation of the neural tube, and we have the neural crest cells. Are these steps clear? Elongation and folding, bending of the neural plate, okay? Convergence into the midline, appearance of the mid-hinge point and the dorsolateral hinge point, and then fusion of the ectoderm of the midline, separation, pinching off, and delamination of cells to become the neural crest cells. All of these are just part of the primary neurolation. So the underlying dorsal mesoderm and the pharyngeal endoderm in the head region going to start signaling the ectodermal cells to elongate into a columnar plate cells, neural plate cells. Okay. So normally up to 50% of the ectoderm becomes neural plate. That's a good proportion. Okay. The neural plate lengthens along that anterior-posterior axis while narrowing. Okay. And that makes the neural plate to form that tube rather than a spherical capsule. Okay. Just because it lengthens. So the neural plate lengths and narrow via convergent extension. And also by doing that, they're going to be intercalating cells into few layers. Okay. And that's how we form that plate. Now, the bending of the neural plate involves the formation of the hinge region. We saw two, the middle hinge that divide the left and the right side of the embryo. So, and this region is made of the epidermal ectoderm that contacts the edges of the neural plate. Okay. That middle hinge cells anchor to the notochord. Yes, and I forgot to mention that it was right here. Here is the notochord in red, right in the middle. And that's how it's anchored. That mid hinge is anchored to that notochord in here. I forgot that. And the two other hinge region are the ones that are going to form that dorsal lateral hinge points. Okay. And they anchor to the surface of the ectoderm and the neural fold. So after the initial furrowing of the neural plate, the plate bends around the dorsal hinge point, and each hinge acts as a pivot that directs the rotation of the cells around it. So they're going to fuse together to the middle. So if I can see now, this I have another one here. Okay, so this is where they continue here. We're going to see in the next slide the different factors are going to be playing a role into how these cells are going to start joining here in this mid-line. Okay, so these movements are changing massively this plate initially seen into what we know as the tube. So what are the different things that play allows them to transform into this incredible shape? So here, our lovely friend, and we want to see how the notochord, that seems such a small structure, directs so greatly all the transformation of our brain and neural tube. So the notochord induces the formation of the middle hinge point. Okay. And this, it does it with a molecule that we have seen again and again and again, Sonny hedgehog, our paracrine factor. Okay, Sonny. Notochord secretes massive amounts of Sonny hedgehog, which in this case will act as a morphogen, because as we go up, the concentration of Sonny hedgehog is decreased. Okay, because as always, diffusion, right? The farther you go, the more diluted it gets. Okay. So what happened is that with the secretion of Sonny hedgehog by the notochord, we are inducing the cells that are just above them, which is at the end will become the floor plate of the neural tube, right? As we see in here, to start elevating. Okay, so without this, we won't be able to lift each of the sides, right? Remember, it's a plate, it's flat. So we need this notochord to induce the hinge point so each side can elevate, okay? And this is with great amounts of Sonny hedgehog. Now, as I pressed too much, as they are going up and they are getting to the midpoint of the embryo, the midline of the embryo, Then we have the dorsal lateral hinge points, okay? And these are induced by another factor that is noggin. So noggin is highly expressed in this upper section and is critical expressed by these neural folds, okay? So as seen in this figure also, we have another element, the BMP. Remember, we mentioned that the BMP in high quantities is really important for the formation of the epidermis, right? So BMPs are expressed by the surface ectoderm, which is in green in here, while noggin in blue here is expressed in the dorsal neural folds. So these are the neural folds in here. So noggin directly inhibits BMP. This is the inhibition process that we see here with this arrow over here. And then the dorsal lateral hinge point develops exactly at that point of interaction when Sony hedgehog is in low quantities, but noggin is in high quantities, okay? Is this interaction understood into Sony hedgehog, BMP, and noggin? Yep, I'm just looking at Annika and Emma. Yes, yes, okay. Okay, so now is the closure of that neural tube. So the neural tube closes as the pair of neural folds meet that dorsal midline of the embryo above, okay? So the two folds will adhere and the cells will start merging to form that neural crest. Then the neural crest cells begin to migrate away, as we're gonna see with the delamination. Okay, and the closure of the neural tube does not occur simultaneously through the embryo. Okay, remember that we're going to have the anterior portion start closing, and then it proceeds cranially to caudally. Okay, so and then the two open ends of the neural tube are called anterior and posterior neuropores. Okay. So we're going to have those neuropores anterior and posterior. And this is something that you're going to be seeing in the lab of neurolation. Now, that closure of the neural tube happened in different ways depending on the animal. So here we have the mouse and we have the humans, okay? And in chicks also is it's a little different. So in chicks, the neural tube closes start at the level of the midbrain and then zips towards each end in both directions, okay? Mammals, even though these two are mammals, typically initiate closure of the neural tube in several places. And this is how we have closure. One is the cranial rachis, okay? And then closure two is here in the midbrain over here. Failure will produce what we call the anencephaly. And then we have three in here. So we will have other conditions. And when it's in the posterior neuropore is when we have the Spina Bifida. So failure to the closure of that neural tube can create different type of conditions. And so these are the neural tube defects, okay? And we have the incomplete closure. So failure to close of the posterior portion is the Spina Bifida. Failure to close the anterior region, either 2 or 3, okay? 2 or 3 is generating the anencephaly, and we're gonna be reading papers about this. Okay, I just need to upload them today. And we also have the closure, the failure to closure at the back, and this is the cranial rachis. And the same happens in humans. So we have the same neural tube defects happening. Okay, now I say that they need to fuse, right? They need to stick together these folds as they get into the formation of the neural groove, and then to separate and form the neural tube. So the separation of the neural tube seems to be regulated by the expression of a molecule that we have already seen, and are the cadherins. Okay, so these are cell adhesion proteins that we have seen in, I think, about 5 to 6 chapters already. Okay, so as we see in this figure, cells that will become the neural tube initially are going to express E-cadherins, because this is the plate, right? So we have E-cadherins are those that are to form the neural tube, okay, at the level of that neural plate in the presumptive epidermis. And E-cadherin expression is stopped at one point, and cells start now to synthesize N-cadherins, okay? And this is so E for epithelial, as we defined it before, and N for neural cadherins, okay? So as they start thickening and elevating, we stop the expression of the cadherins here in the neural plate, and they start expressing the N-cadherins, okay? So the neural tube cannot longer adhere to the epidermis once it is fused here in the midline. So some of the neural tube defects are not rare in humans. Normally 1 in 1,000 live birds will have some sort of neural tube defect. Okay, so it's way more common than we may think. Okay, so as said a couple of the slides before, neural tube defects are caused by incomplete closure of that neural tube. Failure to close the anterior neural tube will create the anencephaly, as that's the example that we see in here. So the forebrain remains in contact with the amniotic fluid and degenerates. You see that the vault of the cranium is not formed in here. And then the brain starts degenerating. The forebrain develops, ceases, and the ball of the skull fails to form. So poor little baby, this baby is not viable. Okay. Failure to close of the posterior neural tube, 0.5. And normally this occurs at day 27. So it's very early in development, right? Will form that spina bifida. The severity of that varies in between people, and it all depends how much of the spinal cord remains open. So some will have just a little hole, and does not affect necessarily the functioning and the mobility of the individual. Some is very severe and they can't walk because the hole is big. So some can go surgery, some surgery is not even an option. So if we describe really this spina bifida for a one, I just, excuse me, I just look at the watch and I saw noon. It's like, what? No, I haven't spoken for that long. Okay, so spina bifida is a neural tube defect disorder, okay, and involving not only the brain but the spinal cord and the protective covering, okay, and in this case it all depends, and all of this, as I say, happen within the first month of pregnancy. And this is once again one of the reasons why I say a pregnancy needs to be planned. Okay, it's not like oopsie. Because a lot of things, day 27 is normally the closure, and here we see we can have issues here. So infants that are born with spina bifida sometimes have an open lesion on their spine where significant damage occurs, and the nerve of the spinal cord has occurred too. So these babies won't be able to move necessarily. So although the spinal opening can be surgically repaired shortly after birth, the nerve damage normally is permanently. So that's result in varying degrees of paralysis of the lower limbs of the individual. And sometimes even when there is no lesion present, there may be improperly formed or missing vertebra, okay? That normally also comes with some sort of nerve damage. Plus the physical and mobility difficulties that these individuals may experience. Most individuals that have some sort of Spina Bifida also have some learning disabilities, okay? Many children with the Spina Bifida have hydrocephalus. Hydrocephalus is an excessive accumulation of cerebrospinal fluid in the brain, which increases the pressure, and normally it will create other type of conditions for that individual. And normally brain can't stand that pressure and the individual will die eventually, okay? There is no cure for this. Cannot be replaced or repaired the spinal cord, right? We can have, however, ways to avoid it. And that's the papers that we want to be reading is this is why pregnancies need to be planned, and the consumption of folic acid is so important. Folic acid seems to be highly correlated with the proper closure of the spinal cord, the neural tube, for the formation of the spinal cord. So it's available. You can avoid it. So in here we have that in humans, neural tube defects can be either due to genetic causes, environmental, or even dietary factors, such as cholesterol, folic acid, vitamin B9, diabetes, obesity, and toxins can all influence human neural tube closure. So you are exposed to so much, right? It is thought that about 50% of human neural tube defects could be prevented if pregnant women to take a supplement of folic acid. Folic acid, we know, is vitamin B12, okay? And it only needs 0.4 milligrams per day. So it's not that much. Now, with age, that's something else, because our metabolism becomes less efficient to use that folic acid. Okay, so young people are most likely to have a good metabolism of folic acid, but older women a little less. So normally for women under a certain age, and when I say certain age, even 30 is considered old to carry a pregnancy. So that's what we're normally going to give about 10 milligrams of folic acid. The Materna pills that you can find in the pharmacy, it has a more or less 10 milligrams of folic acid among a whole whack of protein, vitamins too, that it comes with, okay? Even though that, new research are not necessarily agreeing with this, because it also depends on the metabolic system of females, right? So even though we have a good amount of folic acid, each individual is different, and the conditions that those individuals go to also can affect that metabolism. So it's better to have kids when you are younger than when you are old, okay? There are also some mutations in PAX3, in Sunny Hedgehog, in grainy head, and TFF2 and open brain, and genes that are important for the formation of the mammalian neural tube. And anything that affects any of those, then we're fried. We're done. Okay. So the neural tube eventually forms a closed cylinder that separates from the surface of the ectoderm, and a result of the expression of different adhesion molecules. And in this case, not only the cadherins, but other chems. we allow that closure. So in this image, we're going to be looking at the secondary neurolation. As I say, in most vertebrates, the secondary neurolation occurs in the caudal region of the neural tube. And it involves the making of a medullary cord. That is what we see in this image. That needs to undergo a process that is called cavitation. That is the opening of the hole in the middle of that cord. So here in A, what we have is the medullary cord that is formed at the most caudal end of the cheek tailbone. Okay, so we have a condensation of the mesenchymal cells to be able to form a solid cord. Here in B, that medullary cord starts having an anterior position in the tailbone. So we see that it's completely compacted, more as a cord, right? Then here in C, the neural tube starts cavitating. So this is a little bit of process of apoptosis that is going to happen to hollow that tube that has formed. And then in D, we have the complete lumen is done. And underneath, we see the notochord, how it also helps to induce the fate of those cells. So, as we saw prior in the cheek, this is the cheek embryo development, and this is early brain development formation. Okay, so the Hensen's node, remember that Hensen's node, this little structure over here in the most anterior portion of the embryo. That Hensen's node starts migrating posteriorly. Okay, and as it gets posteriorly, we are closing that tube, okay? So the embryo start closing anteriorly. This is going through primary neurolation in this section, and the epiblast then start emerging in there, and the magic wand group of cells will give rise to that neural ectoderm, and then the paraxial and mesoderm that are going to be on each of the sides, until that here is a more advanced section of it. The Hensen's node is over here. So we're still here in neurolation, while the anterior portion has already start organogenesis. Okay, so in here we're going to see these somites, the formation of the somites. These bulges over here are the somites, which will form the vertebra, as we're going to be seeing later. So these ectodermal cells will form the posterior secondary neural tube that is going to happen over here. Okay, and for this we're going to need the gene SOX tube. Okay. So we're going to have ingressing mesodermal cells, okay, and this will encounter low levels of BMPs, right, because we are at the back portion of this embryo. And we're also going to be using other, that is the TBX6, okay, that will help for the formation of the somites. So not only SOX2, but also TBX6. Now, this anterior portion of the embryo will go to the determination of the anterior-posterior axis specification. So the early mammalian neural tube is a straight structure, right? That's what we see in here. It's like really a tube, right? Early in development, before the posterior region of the tube has even finished forming. As we saw, let me go back. So we are here, we have here the anterior portion start forming and the posterior isn't even ready, okay? So the anterior region start ballooning, as we see here. So start having a little bit of ballooning, and we have very early the three primary vesicles of the brain. The prosencephalon, the most anterior part, that is the forebrain. The midbrain is the mesencephalon, and the posterior brain is the rhombencephalon. Okay, so fore, mid, and hindbrain. Later in development, we have that there is a closure somewhere over here where the spinal cord will start, and that's what allows more liquid to the spinal fluid, okay, to accumulate in this, and allows the expansion of these three vesicles, initial vesicles. So the prosencephalon, the most anterior, will separate into two more vesicles, the telencephalon and the diencephalon. The telencephalon will have the olfactory lobes, hippocampus, and cerebellum, so the senses of the smell and memory and association. The diencephalon, the second part of the prosencephalon, will contain the optic vesicles, the epithalamus, thalamus, and a hypothalamus. The midbrain do not subdivide. It continues to be the mesencephalon. That will be part of the midbrain, which is the temporal regulation, motor control, motivation, and emotional control. And the rhombencephalon will also subdivide into two more vesicles, the metencephalon and the myencephalon. The metencephalon will become the cerebrum and the pons, while the myencephalon will become the medulla, okay, which is the center of the involuntary activities. Medulla oblongata also, okay? So this section over here is really important because this is what is going to control our respiratory, gastrointestinal, and cardiovascular movements, okay? So from a tubular containing prosencephalon, mesencephalon, rhombencephalon, we're going to have the telencephalon, diencephalon from the prosencephalon. Mesencephalon is the only one that does not subdivide, and the rhombencephalon in metencephalon and myencephalon. Pay attention to this because in the slides from the lab, you're going to need to identify the different sections. So well knowing which of them are placed is important for the identification. And caution needs to be made too because as we're going to see, embryos have a curvature, right? So depending on the position of that curvature, you may be looking at telencephalon and metencephalon at the same time. So you gotta be careful into the position and knowing the exact position, more from telencephalon followed by the encephalon and so on, okay? Okay, the rhombencephalon develops some segmental portions that we're going to call the rhombomeres. Called rhombomeres because it derived from the rhombencephalon. Okay? These rhombomeres specify where certain nerves originate. Okay? So the neural crest cells above each rhombomere, so here we have the plate, we have the fusion over here, and the rhombomeres that start appearing in there. So above each rhombomere, we're going to have the formation of the ganglia. Do you remember what ganglia are? Not too much for those that have taken anatomy and physiology. You should know that. So ganglia are simply clusters of neural cell bodies whose axons form a nerve, okay? So this is a group of a cluster of neural cell bodies, not the axons. So the cells within one rhombomere can freely mix, but are associated from cells in other rhombomeres. So it's not that this will be forming a cluster, it's that the posterior part of this rhombomere and the anterior part of this other rhombomere will form a cluster, okay? So each rhombomer has a different developmental fate, Okay, so each rhombomer expresses a unique combination of transcription factors, and we want to be seeing a paper for this, capable of generating a specific neural differentiation. So neurons with distinct fates, as we will see in a couple of chapters. So remember about the ballooning that I was mentioning to go from a 3 vesicle into a 5 vesicle. This is exactly what we can see in here. So here is the closure. Here are all the brain vesicles, and here is the neural tube with the somites forming. Okay. So in chicks, this is in chick, we're normally going to see a volume increase about 30 times between day 3 to day 5, okay, of development. So this rapid expansion is thought to be due to the positive fluid pressure exerted here in the main 3 vesicles because of the closure that we have in here. So there's a constraint in there, okay? And here normally is closed and here is open. Okay, so that section over here is absolutely closed in B, in C, excuse me, here. So once the ballooning has occurred and we have the 5 vesicles, this is 1, 2, 3, 4, and 5, okay? Normally by the end of day 5, we're gonna see that the occlusion opens. Oops, my mouse. Okay, come on. We're gonna need to use the other mouse. This one doesn't go there. So, and then it completes, it goes inside the tube. Okay, and that's what allows for the expansion. The dorsal ventral axis is the second axis that needs to be specified. And we see in this figure that we have that polarization. Okay, so the dorsal region is where the spinal neurons receive input from sensory neurons. So all of this section is sensory, and this is the dorsal part. This is anatomy 101, okay? The ventral region is where motor neurons are. They reside in the ventral region. And in the middle, we'll have what we call interneurons, right? So this is the neurons relay information between the sensory and the motor units. So signals from the environment immediate around the neural tube determine its polarity. So we have normally 2 factors that are going to be playing a role in this. And it will be, what were the 2 factors in the formation of the neural tube? Sunny hedgehog and BMPs in this case. Okay, so sunny hedgehog and BMPs are the 2 main factors that are going to play a role into this. So the dorsal pattern is imposed by the epidermis, which normally we know that it will be secreting BMPs, right? And the notochord. So that's what I say, the notochord, that it seems so little, so transient because it's transient structure, but it's so capital for the formation of the right pieces at the right time. So sunny hedgehog is secreted by that notochord, and BMP that is normally a factor of the ectoderm in there. So in both cases, the paracrine factors induce a secondary signaling center within the neural tube itself. And this is what we have here in this end. Okay. PAX3 and PAX7 define the most dorsal domain. So this is the dark blue in here. It's PAX3 and PAX7. Then we have also NPX6, which is expressed at the bottom, at the floor of the neural tube. So this is what is in red. PX6, PAX6 in here is located into the central region. So wherever is a little more on the greenish color and the transition, that is the PAX6. So there are two regions where we see an overlapping of the factor, the light blue and the yellow, right? And is the gradient between how much Sonic hedgehog can induce this factor and how much the BMP from the above can induce this other factor. So that's the in between the layers. But PAX6 is the green line in here. Okay, so Sonic hedgehog determine the fate of the ventral neuron and is secreted by the notochord in huge quantities here in green. We say that Sonic hedgehog induces the mid-hinge point, right, to become the floor plate of the tube. Okay, and these floor plate cells also secrete themselves Sonic hedgehog. This is why we say that even within the tube, we have an induction of secretion. Not only the notochord induce the cells to become, but once they become the floor, the hinge, they also secrete Sonic hedgehog. Okay, so Sonic hedgehog protein forms a gradient that is at the highest at the more ventral portion, and this is what we see in here, is really dark green, and this is what induces that gradient, and this is where we have these motor neurons in this section. So the different concentrations of honey hedgehog function by causing different group of neurons as we get far away from this notochord and the induction of the mid-hinge point to take different fates. Okay, so the transcription factor activates genes which give different identities. Okay, so we're going to go back to that in a second. So the most closer to the plate are the motor neurons, and as we get farther away, getting into the midpoint in here, then become interneurons. Okay, just because the gradient is too low for Sonic hedgehog. Then the second gradient is the one that comes from above. And those are the BMPs. Okay, so remember BMPs are belongs to the TGF beta superfamily. Okay, those then are secreted by induced first by that ectoderm. Okay, so just as the notochord induced the secondary signaling on the floor plate, the epidermis also induces that secondary signaling from above. So in here we have different factors. So we have BMP4, we have BMP7 also that gonna induce those cells in this section over here. So BMP4 is the main protein that induces a cascade of the TGF beta on the roof of that neural tube. The same thing as the notochord induced the mid-hinge point to secrete Sonic hedgehog. In this case, BMPs will induce the roof of the neural tube to secrete more BMP. Okay, is that clear? Yep, okay. And wherever is in the middle, in here will be interneurons. This is our last slide for today. And this figure shows a model of the dorsal ventral pattern in which the TGF beta and Sonic hedgehog morphogenes act to decide the fate of the cells through the central nervous system, and this into the rostrocaudal axis. Okay, so this is the anterior portion of the embryo. This is the posterior portion of the embryo. Okay. And then we have the dorsal and we have the ventral part of the embryo. In the cranial section, so this is just for you to guide dorsal, ventral, anterior, posterior, and then the paraxial mesoderm. So if we look at the cranial, I'm going to go with this mouse better. In this cranial part, of the neural tube. The brain and the spinal cord normally get their fate directed from the epiblast. Okay. So cells from the posterior part of the embryo are bipotential neuromesodermal progenitors. This is what this NMP means. Neuromesodermal progenitors. Okay. And this will become somatic or neural cells. So here we see how they start going through the paraxial sites, right? From that paraxial mesoderm. Okay. So these neuromesodermal progenitors are maintained by the expression of high concentrations of what we see here, FGFs and WNTs. And this is normally at the posterior part. So this FGF and WNT continues to stimulate these neuromesodermal progenitors to continue dividing and promote the migration. The anterior portion of the embryo has a great gradient of retinoic acid. Okay. And this retinoic acid will inhibit normally the FGF at that portion. So that's how the anterior and posterior portion are determined in this case. Okay. Some mesodermal progenitors at the tail, as we see in here, will enter the neural mesenchyme and become, we see it well in the middle, the prenatal progenitors. Okay, so it's just all, and this section migrates from here and it goes up to here. Okay, these prenatal progenitor cells are susceptible to Sony hedgehog or BMP's signals. Okay, so they're gonna base their position on the floor plate of the roof. So here we go, and goes this end. So then don't stay in here, they migrate into this section. So as these progenitor cells place themselves farther from this signaling of the FGF, they cluster to the retinoic acid, which have higher concentrations in here, and their competency is triggered, and now they're gonna respond to the gradient that we have between Sony hedgehog and the GTF beta, either to become neurons that are gonna be motor neurons or neurons that are gonna be from sensitive pathway. And anyone that is in between, then it's gonna be interneurons. So that is how the cranial posterior portion is determined, posterior with high concentration of FGFs and WNTs, anterior with high concentration of retinoic acid, and as the prenatal progenitors goes and move towards the anterior part of the embryo, then they're gonna be susceptible to the Sony hedgehog or to the BMP. Was that clear? Yeah. Okay. So, and this explains so much as we're gonna progress too, because FGFs and retinoic acid are gonna be mentioned again and again and again, and Sony hedgehog and BMPs also are going to be named again and again and again. So, with that, that was all I have for you today. And I'm gonna see you guys on Thursday to get into how the brain forms, which is one of the most magical aspects of all of us, that what it gives us our own identity and so on. So I'm gonna be uploading some papers later on, okay? And see you on Thursday. Wish you have a good week. Bye-bye, guys. Okay.